an impulse is going through the body, it's going to go from the outside world to the brain and then back out to have that response. As the nerves in the body are collecting the impulse, it's going to want to kind of converge that down into one neuron at some point in time. So we have neuron convergence. That's when we have multiple neurons sending a similar message to one neuron in order to get the message sent. Divergence is the opposite. This is when we're coming from the brain and need to send the message out to several different places. We have neuron divergence. So one neuron is sending the message out to several other neurons. A reflex arc is a safety measure within the body that bypasses the brain. We have a receptor, a sensory neuron, that is picking up some sort of information. It's bringing it to the spinal cord and then back out to the effector without ever going to the brain. This can be done with 2 or 3 neurons. Generally, it's going to have a sensory, maybe an interneuron. That interneuron is not always there, and then having the infector, and never going up to the brain. Reflexes are safety measures. They are safety measures in the fact that if this message had to go to the brain, it would take too long and too much damage would be done to the body. Some examples of a reflex arc are your knee jerk reflex arc. When you hit just below the knee, it causes your leg to kick. The reason why we have this is so we actually can't fall asleep standing up. If you fall asleep when you're standing up, you will actually fall because you don't have the awareness about you in order to sleep while you're standing up. Horses actually have a mechanism built into their knees so that way they can sleep standing up. Us as humans don't have that, so it kind of saves us from falling down. So the knee jerk reaction is to help you from falling down from the standing upright position. The pharyngeal reflex is when you kind of swallow something you shouldn't, it causes you to gag. So if you kind of bypass the swallowing and something gets down to the back of your throat, the corneal reflex causes you to blink or close your eyes when something gets close to your eyes. And then you have the withdraw reflex, that if you touch something hot or painful, you're going to withdraw immediately. So when you touch a hot stove, your hand's not going to sit there and burn. You're going to touch it and you're going to quickly remove that hand or body part from that hot or painful, so that way your body doesn't get injured. Let's take a look at the central nervous system. So we're going to take a deeper look at the brain and the spinal cord. The outer layer of the central nervous system is called the meninges, and the meninges has 3 different layers. If we take a look at this picture here, the top layer here is the scalp, then we have the skull, and here is the outer kind of covering of the brain. The outer layer of the meninges is called dura mater. This is a very thick outer layer covering of the brain. When we dissect our brains, the sheep brains in class, the dura mater will actually have already been removed. The middle layer is actually this here, and that's called arachnoid matter. And the reason why it's called arachnoid matter is because it's a mesh, kind of looks like a spider web. And then the innermost layer is touching the brain directly, or the brain and the spinal cord directly, and that's called the pia mater. 3 layers of the meninges. The most superficial is the dura mater, the middle is the arachnoid matter, and the most deep is the pia mater. The pia mater is directly touching the brain and is a very thin layer along the surface of the brain. The dura mater has actually been removed from the brains that we are dissecting, so we will actually not see that. This is a picture of a brain with dura mater, and you can see that the dura mater has actually been pulled back here in this one section. So this is the thick outer layer of the meninges covering the brain. The spinal cord is separated up into 32 different pairs of nerves. There's a nerve coming out from each space between each vertebrae, and they innervate different kind of pools of areas. The cervical nerves here will generally innervate the upper body and mostly the arms. The thoracic area is going to innervate mostly the chest. The lumbar area is going to do most of the legs. And then the sacral area is going to kind of do that pubic region and the outside down of the outside of the legs and part of the feet. Here's a closer look at the spinal cord. Here we can also see the meninges. We have the dura mater and the arachnoid mater here. The pia mater would be tightly touching the spinal cord itself. We have white and gray matter. We have the white matter here on the outside, and then the gray matter on the inside here. And then we have those nerves kind of branching out on each side of that spinal column out to the periphery. The human brain is going to look a little bit different than what we're dissecting, as that humans are upright, so the cerebellum and the brainstem point downwards, are kind of underneath that cerebrum. And here is a sheep brain, which is what we're going to be dissecting. The cerebrum is here, and the cerebellum and the brainstem kind of go backwards out from that because the spinal column is behind the head as opposed to below the head. So I wanted you to see the shape difference between a human brain and the brain that we are dissecting of the sheep. The cerebrum is the large part of the brain. So this is the cerebrum here. You have two cerebral hemispheres, one on the left and one on the right. This controls most of your daily thinking, and it's separated up into 4 lobes. We have the cerebellum. The cerebellum is located here. In the picture, it's here. And then we have the brainstem. The brainstem is located here. And the brainstem is what connects the brain to the spinal cord. If we are to cut the brain in half, so this is a mid-sagittal section or a medial section of the brain where it's cut straight down the middle, the part of the brain that connects the 2 cerebral hemispheres is called the corpus callosum. So this is where the left and the right cerebral hemispheres come together. This is where the nerves join for the left and the right hemisphere of the cerebrum. The cerebrum itself is made up of all these convolutions. The convolutions, or the gyrus, are these. And an interesting fact, with Alzheimer's, you will have less convolutions in your brain when you have Alzheimer's. That used to be the only way to truly diagnose Alzheimer's, is to take a look at the brain and look at the convolutions, and a diminishing amount of convolutions was indicative of Alzheimer's. The holes that are in between the convolutions are called sulci, or the sulcus. There are deep holes, also known as fissures. So fissures and sulci are the kind of the indentations where the gyrus are the convolutions. So let's take at the different lobes of the brain. The lobes of the brain should be fairly easy to remember if you can remember the skull bones, because they're named after the bone that they're found underneath. So we have our frontal lobe underneath the frontal bone. We have the parietal bone, or the parietal lobe, the temporal lobe, and the occipital lobe. So looking underneath the brain, we have the frontal, the parietal, which is here, you can barely see, the temporal, and in this picture from underneath, you can't see the occipital because it's at the base of the head and the cerebellum is covering it. Taking a look at what each part of the brain does, we have the frontal lobe, and that's most of your problem solving and your emotions, some behavior and personality. Your temporal lobe is for memory and understanding language. Parietal lobe is your math and spelling. The occipital lobe is mostly vision. So a couple association areas that I want to highlight is we have the motor cortex. The motor cortex is this area here, and this has to do with movement. We have the sensory cortex here, which is picking up sensation. There's an area in the frontal lobe called Broca's area, And there's an area back here called Wernicke. And these both have to do with speech. Broca's area is more with the actual formation, and Wernicke's is more of the understanding of speech. The cerebral cortex is kind of the outer layer of the cerebrum, and this is this kind of outer layer of the cerebrum, and this is where most of your thought takes place. The whole 10% of using 10% of your brain is actually a myth. You do use all of your brain, you just don't use it all of the time. So each portion of the brain is used, it's just not necessarily firing all at once. Because different parts of the brain are responsible for different functions within the body, and you're not necessarily having activity in every single spot of the brain at every single moment in time. So you do use all of your brain, just you don't use all of it all at the same time. Within the brain, there are ventricles. Ventricles are basically a space within the brain. The area highlighted in blue are your ventricles. Within the ventricles is cerebral spinal fluid. The cerebral spinal fluid is there for kind of a buffer protection between the brain and the bone that surrounds it.